of you have ever thought about losing weight sometime in your life? Can you show me your hands? Me too. After finishing high school in China, I flew all the way to Michigan to start college. And as many of you might have heard of the freshman 15, for me, it's even worse. Meeting new people and getting into new food, which all make me feel so excited. And eventually, after my first year, I gained 25 pounds. I mean, 25 pounds. I've never been so fat any time in my life. And ever since then, losing weight has always been a priority item to do on my to-do list every single year. But every year, I fail. Why? It's because I simply, with all the homework, all the lab work to do, I simply don't have enough time to hit the gym 5 times a week. Or with my passion and love for food, it's even harder for me to keep a diet like this. So is it impossible for people like me to lose weight? As I entered graduate school 4 years ago here at Caltech to study cell biology, I have this little secret hope that using my knowledge towards understanding how cells work, I could understand better with how fat are aggregated and decomposed within our body. Maybe I could come up with a solution that help people like me to lose weight. What if we could simply reduce the number of fat cells in our body? Or even better, what if we could turn our fat storing cells into fat burning cells? This sounds like a wild idea, but it might be true. And here's why. It turns out we have more than one type of fat cells in our body, which we will call adipocyte. So the most commonly known and most abundant one is called the white adipocyte. And all that they do is to store the fat. They are the culprits that give me this awkward love handle when I'm wearing my swimsuit, and that's why I call them the bad fat. But in the meantime, we have another type of fat cell, which we call the brown adipocyte. Unlike the white, the brown can actually, instead of storing fat, but start to burn fat. The brown adipocyte can utilize fat as the energy source to generate heat. But for humans, unfortunately, the brown adipocyte only exists in infants. For newborn babies, they are the major organ to keep the baby at a stable temperature because they can generate heat. But as we grow into adults, we gradually lose all of the brown fat. So in short, the brown adipocyte are the good adipocyte, but only exist in infants, not adults. Now comes the third type of adipocyte, which is called the beige. And based on its name, it's easy for you to imagine that it is just in between the white and the brown. And that's exactly what they do. It is the intermediate form of adipocyte, which first they will co-develop with the white adipocyte, which are the bad fat cells, first to store fat. But upon some environmental stimulation, such as coldness, the beige adipocyte can actually be induced and activated and start to behave just like the brown, which are the good fat. So they could, instead of storing fat, they could start to burn fat. Now this beige adipocyte sounds like our solution, right? Just imagine if we could induce more white fat into the beige fat. In other words, to turn more bad fat into good fat, we might be able to finally lose weight. So what's causing the difference between those two types of adipocytes, and what could be driving the change between those two adipocytes? One of the major differences that exists is that the number of mitochondria. Briefly, the white adipocyte has very few mitochondria per cell, but for the good, the beige and brown adipocyte, they have a lot of mitochondria within their cell. So now we can turn the question basically into how can we change the number of mitochondria within the cell in order to change the type of fat cells we have. But first thing first, what are mitochondria? A lot of you guys might already get lost. Like, what are they? Mitochondria are those tiny organelles that work in our cell. They are very small in terms of size, with the diameter of about 1% of the diameter of your hair. Besides the small size, their biological functions are extremely vital for the cell's survival. Because just imagine your cell as a factory. The mitochondria are the powerhouse of the factory. They provide everything, all the type of energy you need to drive your cell to work. So all the energies that you intake from the outside, for example, the sandwich you just had, or the orange juice you had this morning, all of the energies will be eventually transported to mitochondria and then be converted from the chemical energy our cells can't use into the biological form of energy that our cells can actually use, which is we call ATP. So let's get back to the number change of mitochondria. How can we change the number? Well, it sounds simple, right? If you want to have more mitochondria, more powerhouse of the cell, you just build more, meaning you just generate more mitochondria. And if you want to have less, you just degrade more, meaning you just break them down and get them disappeared. And in this talk, I will mainly focus on the degradation of mitochondria, which will cause the number decrease, so that you can simply imagine you could if you have mitochondria number decrease, you will have the fat from the good fat revert back to the bad fat. And our cell has a very regulated mechanism to degrade the mitochondria, and here is why. Showing in green is a mitochondria that is ready to be degraded. Once it's ready, it somehow signals to the cell, release some signals to the cell, letting the cell know, I'm ready to be degraded. And then the cell will make a decision that, oh, okay, I'm going to eliminate this particular mitochondria. And by sending a team, like something like here, a team to recognize this targeted mitochondria, this team of molecules will gradually wrap this mitochondria up and isolate it from the surrounding cell. Eventually, this particular mitochondria will be degraded within this vesicle. And that's how we get our mitochondria number decrease. So in order to study all these processes, like the number decrease, like our fat cell type change within the cell, how we do it, as cell biologists, we can't just recruit people to the lab like, oh, I'm interested in how the mitochondria function in your cell. Can you let me dissect you up and then take a look? I mean, that sounds a little bit dangerous, right? So what we do here is actually we use something like this. So we isolate the cells from either mouse or rat or even human patients. And then with the isolated cells, we grow them into this little plastic container with different growth medium. And in our lab, we're able to grow the different type of adipocyte with different growth medium so that we can have the white adipocyte, the bad adipocyte, and the good adipocyte, which is the beige. And by giving them different stimuli or withdraw the stimuli, we can actually make the white change into the beige or the beige back to white. And this is, and then to visualize. So now we want to visualize, oh, how mitochondria work within the cells, right? To see that, I made a construct which will specifically label different mitochondria within the cell. And now you can see we can label the healthy mitochondria with green and red, and the degrading mitochondria with only red. This will tell us what are the portion of mitochondria that are healthy and what are the mitochondria that are being degraded. So what's going to be the number change exactly happening in the cell. So here's just an example of what we daily see on under a microscope. Here is like a intact cell. It's what your fat cell looks like. Those little holes are actually actual fat within your cells. And you can see that as I increase the contrast, you can probably see the healthy mitochondria with the green fluorescence and the degrading mitochondria with the red. And this is actually a cell that are undergoing this mitochondria dense condition to a mitochondria lacking condition, in which case you will see a lot of cells, a lot of mitochondria being degraded. So with this system set up, we're hoping that by understanding exactly how mitochondria decrease or increase within our fat cells, we would potentially have a way to manipulate the number of good versus bad fat cells in our body to lose weight. And hopefully someday I could be able to achieve this goal. So thank you.